Hello everyone, welcome to the ITILS Academy. Today we are going to discuss another topic that is your method for detecting the pathogenic microorganism and their toxin. Okay, we all know this pathogenic microorganisms are what? Pathogenic microorganisms, they are what? They are capable of causing disease into a human being, capable of causing disease into a human being. Okay, so it is very essential that we have to detect a method so that we can analyze which type of pathogenic microorganisms are present and along with it, which type of their toxin. Okay, we all know that this microorganisms are either pathogenic or either spoilage. Now, what is the role of this spoilage microorganism? Spoilage microorganisms secrete their toxin. Okay. And due to which the food are spoiled and secrete their toxin and the food are spoiled and along with it, it become a toxic for the consumption of the human being. Okay. So we have to discuss both the method. Okay, both the detection method for the pathogenic microorganisms and for the spoilage microorganisms of their toxin which release into the like product. Okay, why we it is important for the purpose of food safety. If we don't know which type of microorganisms are present into the food product, then we cannot eliminate it. So before we have to elimination any type of this microorganism, we have to know that what is the characteristic of that particular microorganism. It is very important. So here we are going to discuss the method for detecting the pathogenic microorganisms and their toxin. So let's start today's lecture. Now see the introduction part. Identification and characterization of microorganism is a key part of the management of the food safety and quality, tracing contaminant and troubleshooting problem like your spoilage. Okay, so for the purpose of food safety, for the purpose of food quality, for the purpose of tracing contaminant, okay, or troubleshooting problem like your spoilage. For all these purpose, this identification of the microorganism and after identification, the characterization of this microorganisms are very important. Identification and characterization, both are very important. Okay. Now, the first one is your identification of the microorganisms. It provides the name of the microorganisms, like from which genus it is belong, from which species it is belong, and it will help in determining whether it is safety or spoilage concern. Okay, it is important for the determination of this microorganism for the safety purpose and for the spoilage purpose, and is likely to be a heat resistance. Okay, maybe some bacteria are what? They are a heat resistant. So how we find it? By identification of that microorganisms. Now the next is your characterization of this microorganism. This group together organism that share a similar DNA fragment, pattern or a antigenic profile, which assist the tracking or tracing contamination. That is the characterization of the microorganism. organism. Now the next is your molecular detection of pathogens. So here when we how we detect the pathogen with the help of molecular. Okay, so here we discuss the molecular detection of the pathogen that is called a molecular microbiology. So this molecular detection of the pathogens are totally new, dynamic, and progressive spin-off of classical microbiology. Okay, it is the spin of the classic microbiology, and it is totally new, dynamic, and progressive. And it also play a very important role in those clinical situations where a standard microbiology produces suboptimal result or completely failed. Okay, so this uh, this uh, this molecular microbiology are play a very important role in those type of the clinical situation where our standard microbiology are failed. Okay, this what is the standard microbiology which rely on a successfully cultivation of a potential pathogen. Okay, or this molecular approach are also important for the identification, quantification of the microorganism in a diagnosis of the infection or the different type of foodborne illness by using a molecular microbiology. Okay, broadcast range are also available for testing and tailor-made packages. So this will come under a molecular microbiology. Okay, now the next is your identification of the microorganisms. How we identify the unknown specimens. What is the role of? Okay, so the method microbiology use fall into three categories. So for the identification of the microorganisms, we have a three category. One is your phenotypic, then immunological, then genotypic. Okay, so just genotypic is totally based upon a genetic technique. Okay, and this immunological are based upon a serological analysis. Serological analysis. And phenotypic means that morphology. So by this three method, we can identify the microorganism. Now the next is your phenotypic method. This phenotypic method, it's totally a old-fashioned method. Means that it is used in a old time, old-based method via biochemical, serological, morphological are still used is are still used in identification of the microorganisms. So in the place of this phenotypic method, we can use a biochemical. Sorry, this phenotypic method is old-fashioned, but in phenotypic, this biochemical method, serological and morphological method are still used for the identification of this microorganism. Phenotypic method like your microscopic morphology, which include the combination of the cell, shape, size, gram staining, acid-fast reaction, special structure like endospore, granule, and capsule, which can be used for the identification so that it will give an initial presumptive identification. Okay, now the next is your macro in phenotypic method. First of all, we are going to discuss about the macroscopic morphology. Okay, this macroscopic morphology are trait which can be accessed with the naked eye. Okay, this is a trait and it can be accessed by our naked eye. Macroscopic means that the size are larger, like the appearance of the colonies, including its texture, shape, pigment, and even the speed of the growth and growth pattern in a broth. Physiology or a biochemical characteristic, okay, which included traditional mainstay of bacterial identification, which include enzymes like your catalase, oxidase, decarboxylase, even the fermentation of the sugar, capacity to digest or metabolize the complex polymers which are sensitive to the drugs, and it can be used in the identification. Now the next is your immunological method. Okay, so after phenotypic method, we are discuss the next method, that is your immunological method. Immunological methods are involve the interaction of the microbial antigen with the antibody, and this antibody are produced in a host immune system. Okay, so the antibody which are produced in a host immune system, their interaction with the microbial antigens are involved in a immunological method. Testing for a microbial antigen or the production of the antibodies is often easier than the test for the microbe itself. Now, different lab kits are also based on these techniques, which are also available for the identification of the microorganism. Now, the next is your ELISA. In immunological method, one method we also use, that is your ELISA, which means that enzyme linked immunosorbent assay. This ELISA become a widely used serological test for the antibody or a antigen detection. Okay, this test are also involved in the link between a various level enzyme, either they are antigen or either they are antibodies. Okay, the enzyme which are used in ELISA, like your alkaline phosphate for oxidase and beta galactosidase. Okay, during the indirect ELISA, this antigens are trapped between the two antibody molecules, and it is called a sandwich ELISA. For the detail of this ELISA are already uploaded in our account. You can go and check it and see the video related to this topic. Okay, now the next is your here how we do this ELISA. The specimens is added to a well with attached AB. Now, if the antigen is present, it will attach to the antibody. Suppose that this is your specimens. Now here, when we added this specimens into a well, which is attached to a antibody, okay, if there is a antigen are present in this specimen, it will attach to the AB, that is your antibody. Now we wash it so that the unbound material can be removed. And again, we add a secondary antibody with a conjugate enzyme. Now, the second antibody is specified for the antigen, and then we added a substrate which react with that enzyme so that it will give a colored reaction. Okay, this ELISA tests are used for the detection of the Staphylococcus aureus, E. coli, and Salmonella. Now, the next is your, there is a problem with the traditional method. Okay, now see, we are going to discuss what is this problem. Cultivation based method insensitive for the detecting the some microorganism. Cultivation based method limited to a pathogen with a known growth requirement. Poor discrimination between the microbes with the common behavior features. It is failure to detect the infection which is caused by a uncultivated microorganisms or the organisms that are failed to elicit a detectable host immune response. Visual appearance of the microorganism is a non-specific example of some failure with the traditional approach, like the detection of the slow microorganism take a week, like your tuberculosis. A number of visible microorganisms cannot be cultivated, like your Whipple bacilli. Disease presumed to be infection remain ill-defined with no detected microorganism. Example, abrupt fever after a tick bite. Okay. Now the next is your identification. Now, identification of microorganisms with the help of genotype method. So here we are going to use another method, that is your genotype method. Now, just genotype methods are involve um, identification of the identification of the microorganism with the help of genetic material. Okay, with the help of just this genetic material, what should we do? We examine the examine the microorganism. Okay, has a revolutionized bacteria identification and classification. Genotype methods involve examining the genetic material of the organisms and has revolutionized bacterial identification and classification. Okay, now there are different methods which are included or come under the category of just genotype method, that is your PCR. PCR means polymerase chain reaction, okay, which included RT-PCR, rapid PCR, use of nucleic acid, RFLP, and plasmid fingerprint. Increasing genotype techniques are becoming the sole means of identifying many microorganisms because of its speed and accuracy. Okay, because of its speed and accuracy, these genotype techniques are become a more more important method for the identification of the microorganisms. Okay, now this genotypic methods of microbe identification are included use of nucleic acid probes, PCR, RT-PCR, rapid PCR, nucleic acid sequence analysis, 16S RNA analysis, and RFLP, that is your restriction fragment length polymorphism, and plasmid fingerprinting. Okay, so with the help of this genotype method, we identify the microorganisms like your nucleic acid probe. With the help of nucleic acid probe, PCR, RT-PCR, and RAPD-PCR, both nucleic acid sequence analysis and 16S RNA analysis, RFLP, that is your restriction fragment length polymorphism, and plasmid fingerprinting. Now the next is your application of molecular method in food industry, detecting and identifying specific genes, that is your genetic modified food, application to a food authenticity and legislation, detection of microbial contamination of food, species identification, detection of food constitute, that is your ingredients or contaminant, detection of antibiotic, pesticide residue, etc. Halal and kosher certified. Okay, so this molecular method which are used in the food industry for first application of this is what detecting and identify the specific genes and when our genetic modified foods are manufactured, and even it is the application in a food authenticity and legislation, and it also help in the detection of the microbial contamination in a food, species identification, and detection of the food constitute, that is your either it is ingredients or either it is the contamination, and detection of the different antibiotics, pesticide residue, etc. Halal and kosher certification. Now PCR, polymerase chain reaction. Okay, so PCR, the video related to this are already uploaded in our account. You can go and check it. What is this polymerase chain reaction are? So whole video are related to this are already uploaded. You can check it. This is also PCR, PCR. It is also about the PCR, PCR, PCR. Okay. Now, plasmid fingerprinting. Plasmid fingerprinting identify the microbial species or similar strain which is related to a strain, often contain a same number of plasmid with the same molecular weight. Okay, this plasmid of many strain are used for the identification of the species, that is your E. coli, Salmonella, Campylobacteria, and Pseudomonas, which is demonstrated that this method is more accurate than a phenotypic method. Okay, like your biotyping, antibiotic resistance, pattern, phage typing, and stereotyping. Okay, now the next is your this procedure are involved, bacterial strain are grown and the cell lies and harvested. Then this plasmid are separated with the help of agarose gel electrophoresis, and this gel are stained with the ETBR and the plasmid located and then compared. Okay, so this is all about the different method for the identification of the pathogenic microorganisms and even the toxin produced by the microorganism. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. For further query, you can ask your query to the helpline number that are already mentioned in your slide. Visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.